All right, this video is going to supplement some of the Punnett Squares videos that you guys are watching. This is something that we learn in AP Bio that we don't learn in Baby Bio. We're going to talk about dihybrid crosses. And there are a couple traits I'm going to look at here, and we're doing dihybrids here with just straight up autosomal dominant recessive traits. Okay, so these are traits that don't follow any of the funny inheritance patterns. That's what I'm going to look at here. That doesn't mean that that's what College Board is going to do. So I'm going to show you the technique that you can expect that College Board might mix a, say, an incompletely dominant and an autosomal dominant trait in a dihybrid cross. So you need to be prepared for that. All right, let's go ahead and focus first on the technique. So we're going to start by looking at two traits, and this is a dihybrid, meaning we're looking at two traits in the same Punnett square. All right, so we're going to look at handsome Mr. Hemsworth here, and you can see that his hairline, if you look, it goes down into a peak like this. This is known as a widow's peak, and that is a dominant allele. A straight hairline like the one that you see here is recessive. So widow's peak is dominant, so I'm going to give that a big W, okay, for widow's peak. Okay, and then the straight hairline, I'm going to make, you really have to, with W's, make the lowercase look different from the uppercase. All right, so that's the first trait that we're looking at, widow's peak versus straight hairline. The second trait we're going to look at is Hitchhiker's thumb, and in Hitchhiker's thumb what you can see is the thumb bends back up almost 180 degrees, and in some people it does bend back 180 degrees. That's dominant to having a more straight thumb. Now notice it's not completely straight, but certainly the tip of the thumb does not bend completely back. So again, here, Hitchhikers, which is dominant, we're going to call Hitchhikers. Okay, and then lowercase h we're going to call straight. And forgive the text lingo here in my in my straights. It's better. My handwriting isn't so hot, so at least you can understand that. All right, so the first thing that we have to do when we're dealing with these, if we're crossing somebody with Hitchhikers and widow's peak who's heterozygous for both traits, with another person that has the same genotype, how do we deal with this? And the first thing that I always tell students to do is pretend you're in math class and that these are separated by parentheses, right? And in math class, when you have terms that are separated by parentheses, you know that what you've got to do is FOIL them, right? And if you were taught the box method works as well. So the FOIL method says we take our first and put them together, then our outer, then our inner, and then our last, right? So we're going to FOIL this to find the gametes like we would normally. So our first letters are big H and big W, okay? So and again, what we're doing here is just via independent assortment of chromosomes during meiosis, you're going to get these alleles mixed, right? They're going to get, the gametes that we're creating should be haploid, so they won't have all four of these alleles in them. They're going to get one H and one W, so we're trying to figure out all the possible combinations of H's and W's here. Okay, so the first gamete from either parent would be a big H and a big W. The second gamete, the outer gamete, right, the one that's from the outer, would be a big H and a little W, right? The inner one would be a little h and a big W, and the last one is a little h and a little w. So these are the gametes. Now, normally you would do this twice, right? One for this parent, one for this parent, but because we know that both parents are the same, we're going to call their we know that they're going to have the same gametes. So what I'm going to do now is I'm going to put up a Punnett square using the gametes that we've just created. And this is going to take some paper, okay? And giving yourself as much room as possible, especially if you're like me and you're not the best handwriter, giving yourself as much room as possible is going to be important. Now notice this is a huge Punnett square. It's a 4x4 Punnett square now. The reason why monohybrids are 1x1, or 2x2 Punnett squares is because you have only two possible gametes for each parent. Now we have 4 gametes. And when you fill this out, you're just putting it back together. Put the H's back together, put the W's back together, okay? And here's our H and our W's, because remember we're making offspring here, so our offspring would be diploid and not haploid, right? So here we go. And go slowly with this. Like I said, just give yourself plenty of paper and go slowly so you don't make mistakes as you go. There we are. So, and these are my least favorite problems to do. And handwriting, whoops, see, there's my first mistake. I almost made that, let me erase that here. Made that H incorrectly. And, you know, I'm being very careful to make sure that I put the dominant first and then the recessive. But you certainly, if you don't do that, it's not the end of the world. If you worked in a genetics lab, they would expect that you would be able to do that. All right. Just continuing on. Okay, and we're almost there. And you can see why this takes a lot of space. Okay, so there was our square. That's what we've got. I'm going to give myself a little space here. Because the most evil thing about these Punnett squares is getting the ratios from the Punnett squares. That's the worst part about this. So what we've got to do typically, right now I'm just going to focus on a phenotypic ratio here, okay? Obviously you could do a genotypic ratio, right? But for now, I'm going to just focus on the phenotypic ratio, okay? There are 4 possible combinations that you can get. You could get someone who has Hitchhikers and Widow's Peak, right? Both of those, Hitch and Widow's, right? In order to have Hitchhikers and Widow's Peak, you need at least one big H and one big W, right? So what we're going to do, I've got color available to me here. So I'm going to, every time I see a big H and a big W in a square, I'm going to put a red dot in that square, okay? So if I see Hitchhikers and Widows, a big H, right, and a big W, I'm going to put a dot. So I'm going to put a dot here, I'm going to put a dot here, dot here, dot here, not there, here, not there, here, 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 not there, not there, not there, not there, not there. If I've done this correctly, there should be 1, 2, 3, 4, 5, 6, 7, 8, 9. I ended up with 9 of those, so I have done that correctly. All right, so our dominant, somebody who's expressing the dominant, in other words, who has Hitchhiker's thumb and Widow's Peak, there would be 9 of them out of this cross, okay? All right, so the next possibility is that you have Hitchhiker's thumb, but a straight hairline, right? So that's the possibility. Well, to have Hitchhiker's thumb, you need a big H, but then the straight hairline would be a little W, okay? So as long as there is a big H and all little W's, I'm going to put a blue dot in the box. Okay, now the beauty of using this method for counting is, if I've marked a box already, I know I don't have to count it. So I'm looking for a big H and lowercase w's, like this one, right? This one counts. Not here, not here. This one counts. No, and no on the last one. Whoops, I'm sorry, I shouldn't have put a dot there. There we go. Okay, so now all I have to do is go back and count my dots again, right? And I've got 3 blue dotted boxes. Okay, the next possibility here is to have straight hairline and widow's peak, right? So to have a straight hairline and widow's peak would mean you'd have all lowercase h's and one big W. Okay, so again, I'm going to put a green dot if I see that, and that. Okay, this last box is not that case, so we have 3 of these. And then finally, the last possibility is to be straight and straight, right? Have a straight hairline and a straight thumb. There's only one box left, and that's this little guy here. That's our ratio. 9 to 3 to 3 to 1. Now, again, I'm not going to waste time in this video counting the genotypic ratio. You certainly could. You could break it out and figure out all the possible genotypes. I doubt, especially on this year's AP exam with everything being so short, that they would do that. I could see them asking you about phenotypes, though. Okay, so this is how you do dihybrids. They're tedious, they're not fun, but notice they're also not hard. Okay, so this is not a difficult thing to do. All right, now that you've suffered through all of that, there is a little cheat that you can use. And the reason for the cheat is because occasionally, if somebody wants to be really, really mean, you could get a trihybrid or a tetrahybrid or whatever. You could actually just keep on going, and that would be crazy trying to make a Punnett square for something like that. So we are not going to Punnett square something ugly like that, but I'm going to show you a little technique with the one that we just did, right? With our cross of complete heterozygotes for both of these traits, right? Okay, so this is our cross. Okay, so here's our cheat. And remember, you can do this with tri, tetra, penta, hexa hybrids as well, as many as you want. What you do in this case is take these alleles separately. So I would do a Punnett square for the H, right? Whoops. Right here, okay. And this is easy, right? This is just a plain old monohybrid cross, right? So here we go. Okay, and then I would do a Punnett square separately for my W trait. I'm trying to make the big W's extra pointy here. Okay. Oh boy, hold on, sorry. Made a mistake. Okay. Okay. So now we've got our 2 traits that have been dealt with separately. We've dealt with the H over here, right? So there's our crosses of the H's, our crosses of the W's are here, right? All right, so now what we do is we say, okay, if you want to know who is Hitchhiker's with widow's peak, right? Who's got Hitchhiker's thumb and widow's peak? You look at this and you say, okay, what individuals on this Punnett square have Hitchhiker's thumb? And if you look and you count, you've got 1, 2, 3 with Hitchhiker's thumb out of 4 with Hitchhiker's, right? Okay, and you have only 1 out of 4 with a straight thumb, right? Okay, so to find the Hitchhiker's thumb, you've got a 3 out of 4 here. And again, with this square over here, 3 out of 4 individuals here have widow's peak, right? But only 1 out of 4 has a straight thumb. So you multiply 3 out of 4 times 3 out of 4 gives you 9 out of 16, right? So 9 individuals out of our 16 would have both Hitchhiker's thumb and widow's peak. What about Hitchhiker's and straight? Right? Hitchhiker's thumb, that's our 3 out of 4. but to have a straight hairline would be 1 out of 4. Okay, so now we've got 3 out of 16. And again, straight with widow's peak, right, would be 1 out of 4 times 3 out of 4, which gives you 3 out of 16. And then to be both straight and straight, whoops, straight for both would be 1 out of 4 times 1 out of 4, or 1 out of 16. But if you look what we just did. Our ratio now is 9 to 3, right? Here's our 9, here's our 3, to 3 to 1. We just got the 9 to 3 to 3 to 1 ratio without doing all the work of that dihybrid cross Punnett square. Okay, so all you do if you want a shortcut to the dihybrids is to take each of the traits separately, do a separate Punnett square for those, right? And then multiply the probabilities of each phenotype, if you're asked for a phenotypic ratio. Multiply the probabilities of each phenotype on the square, hitchhikers and widows. Multiply them together to get what you would get if you had done the full-on dihybrid cross. And like I said, if you do, if there's a trihybrid, then instead of doing 2 little Punnett squares here, you'd add a third for whatever the third allele is, and so on. All right, the last thing that I often get asked, if you do have to do, for whatever reason, if you're told you have to do the full Punnett square, one thing to consider is, what if you have a situation like this? Maybe somebody is heterozygous, right? But then you're crossing them with somebody who's homozygous. Well, whoops, let's make that W correctly here. You're crossing them with somebody who's maybe homozygous for both traits, okay? So now, when you do the gametes here, you're only going to get little h, little w repeated 4 times, right? So you know that this is going to be big H, big W over here, big H, little w, little h, big W, and little h, little w, right? But the question students always ask is, can I just do a Punnett square that looks like this, rather than keep on repeating the big little h, little w all the way down the line? And the answer is yes, you can do a Punnett square that looks like this. It shortens your Punnett square, and College Board, again, may do this just to, ooh, this should be a big H, sorry. They may do this just to make your life a little easier, especially this year since we've got a short period of time to answer the questions, right? But you could actually fill out this Punnett square this way, right? And just do a 1/4 Punnett square to do that. Just when you do count your ratios on this square, know that your ratios will be reduced, okay? It's really only a problem in cases of multiple choice questions where you're expecting there to be a ratio that has 4 in the denominator, something over 4, because you've done a 1/4 Punnett square with 4 possible offspring. But remember, in reality, this square would have 16 possible offspring, so you might have to multiply this by a factor of 4 to get an answer. But again, if it's an FRQ like it is this year, they probably would accept it in reduced form or in its form out of 16. So I hope you found that helpful, and I hope you enjoyed the little cheats.